Well, hello, my fellow pre-meds. Welcome back and congrats on making it to the very last video in this biochemistry playlist, amino acid metabolism. And because amino acid metabolism is a very light topic, I thought I'd include a slide on bringing it all together where I discuss fatty acid, amino acid, and carbohydrate metabolism. Again, in one very short slide, but I think it's going to yield you a lot of points if you thoroughly understand the timeline of when each macromolecule is broken down as the primary energy source. But before we get started with this slide deck, I did want to comment on a few things. Some of you may be asking yourselves, why haven't we discussed Western blotting and some of the other lab techniques as part of this biochemistry playlist? And that's because I've decided to make a separate slide deck or a separate playlist for lab techniques because they span multiple disciplines, including biochemistry and organic chemistry. So I'm going to make a separate playlist for lab techniques, okay? And then another thing I wanted to address is that some of you may have wondered why in the lipid metabolism video I didn't include a discussion on LDL, VLDL, HDL. I think these fit better into physiology, and therefore I'm going to include them in my biology playlist when we discuss the endocrine and the digestive system. So without further ado, let's get started over here. Okay, so the whole process of breaking down amino acids is obviously going to start by breaking down proteins. Again, I don't want to focus too much on digestion, more on that when we talk about the digestive system, but essentially you're getting a protein, like let's say hemoglobin over here, and then you're taking one of the subunits and opening it up over here, so you have your primary structure where you have the amino acids joined to one another via these peptide bonds. And then essentially what you're doing is breaking these peptide bonds with special enzymes, including pepsin and trypsin. Again, more on in the biology playlist on that. But for our purposes over here, we get this amino acid, and then we want to break it down further. So let's see how that's done. So you can see right here that the primary fate for amino acids is going to be to recycle them, essentially use them to make more protein. So you break a protein down, you make another protein. But in cases where you have severe fasting or intense starvation, you're going to use them for other reasons, such as energy purposes in the Krebs cycle, or you're going to use them to synthesize glucose via gluconeogenesis, and these are specifically the glucogenic amino acids, as we'll see in a bit. Another alternative fuel source that you can form are ketone bodies, and these are specifically the ketogenic amino acids. But importantly, notice how all the Krebs cycle intermediates do not contain a nitrogen atom, glucose does not contain a nitrogen atom, and ketone bodies, likewise, do not contain a nitrogen atom. But we know that all amino acids contain an amino group. So where does that amino group go? Well, it gets removed in processes known as transamination and deamination. You don't need to know too much about these for the purposes of the MCAT. Just know that that amino group does have to be removed in the process, and we'll see how that happens in a bit. Okay, so this is the overall flow. You can see you go from an amino acid, you remove the amino group, you end up with a keto acid, that enters the Krebs cycle, or gluconeogenesis. And you can see right here, I have aspartic acid. When you deaminate it, when you remove this amino group, it's essentially oxaloacetate. Oxaloacetate can then enter the Krebs cycle or be one of the precursors for gluconeogenesis. So that's how you enter the Krebs cycle or gluconeogenesis. Likewise, if you get glutamic acid and you deaminate it, you convert it to alpha ketoglutarate. Again, alpha ketoglutarate, one of the intermediates of the Krebs cycle, so it can enter the Krebs cycle. Now, when it comes to that amino group, you've removed it either through transamination or deamination. Well, it turns out that NH4 ammonium is toxic to the body, so it has to be removed somehow. Well, it undergoes the urea cycle, and I've written out the cycle over here, not because you have to memorize it, but hopefully so that you feel good that there's at least one pathway that you don't need to know for the MCAT. So you'll have to know this for med school, but not for the MCAT. So once that ammonium group undergoes the urea cycle, that it will be converted into urea and then excreted in the urine, and that's how you get rid of that potential toxin, which is ammonium right here. Okay, this is just a Venn diagram highlighting the different types of amino acids in terms of glucogenic versus ketogenic. So the easy way to remember this is that only leucine and lysine are exclusively ketogenic. The rest are all glucogenic, but then you have some that overlap. How are you going to remember the ones that overlap? Well, first of all, recall we have 3 amino acids that are aromatic from our amino acid video. Those include phenylalanine, tyrosine, and tryptophan. And these 3 overlap between both glucogenic and ketogenic amino acids. Add to that isoleucine, so like its sister leucine, it's also ketogenic, but then again, it also overlaps with the glucogenic ones. And then finally, you have threonine. The way you can think of this is that tyrosine, one of those aromatic amino acids, has an OH in its side chain. Threonine also has an OH in its side chain, so it's also both ketogenic and glucogenic. Importantly, while serine also has an OH in its side chain, it is only glucogenic. All right, as promised, now let's bring it all together by discussing this graph, which explains when the different sources of energy are the primary one used by the body. So you can see after you've just eaten, so right after your meal, you're pretty much exclusively relying on the glucose in your blood from that meal. As that starts to dip down, you want to start to rely on your glycogen stores. So the sugar that was previously stored as glycogen, you start to break down glycogen and rely on that. That sort of peaks from 4 to 6 hours. But then if you continue your fasting period, that'll start to dip as well. And then at 12 hours, you really decrease the amount of glycogen you have, and you want to start relying on fatty acid breakdown and gluconeogenesis. So you can see you start breaking down your fatty acids and you start to make glucose via gluconeogenesis. Now, importantly, one of the precursors for gluconeogenesis is amino acids. So it would be unwise to continue the process of relying on gluconeogenesis as your source of energy, because you're going to start to break down your muscles to get proteins, and we don't want that. I'd like to keep my muscles, thank you. So what you'll see here is that while it is true that fatty acid breakdown and gluconeogenesis are increasing simultaneously, at some point you'll see that gluconeogenesis dips down and you start to rely pretty much exclusively on your fatty acid stores, which makes sense because fatty acids are the largest reserve of energy in your body. And you'll see as you continue to increase the breakdown of fatty acids, the concentration of ketone bodies starts to increase. And that makes sense because when you break fatty acids down, you're getting acetyl-CoA. And recall that acetyl-CoA cannot be transported across compartments because that CoA part of it is a large molecule. So it's not easily transported across membranes. So the way you shunt it out, or you send it out to different compartments, to different parts of the body, is by converting it to those smaller molecules known as ketone bodies. And once it's in the location of interest, it can be converted back to acetyl-CoA to be used in the Krebs cycle and generate some energy. Now, importantly, let's look at what happens at the 24-hour mark. You can see that while you have gluconeogenesis and ketone body synthesis occurring simultaneously, you'll see that at 24 hours, the ketone body concentration is still kind of low in the blood, and the amount of energy you're deriving from ketone bodies is still kind of low. And what's happening at this point is all of the ketone bodies that are being synthesized are being taken up by skeletal and cardiac muscle. And that's because it turns out that skeletal and cardiac muscles are very good at consuming ketone bodies, so the brain never gets a chance to pick up any of the ketone bodies in the bloodstream, and that's why we still have gluconeogenesis running rampantly, because we want the brain to rely on some sort of energy source. However, again, as you continue in that intense starvation period, the brain does not want to rely solely on glucose, because at this point you're going to have to start breaking down proteins and amino acids to synthesize glucose. So that dips down and ketone body concentration starts to increase in the blood to the point where no matter how much ketone bodies, no matter how much concentration of ketone bodies the skeletal and cardiac muscles consume, there's still enough left over for the brain to use. And in fact, at that point, like 72 hours plus, 75% of your brain's energy source is coming from ketone bodies. So that way you spare your muscles and your proteins because you don't need to perform gluconeogenesis. Okay, so these are some knowledge checks over here. Again, if you have any questions, reach out to me and I'll clarify these questions. All right, congratulations on finishing the final video of our biochemistry playlist. I really hope that you found these videos to be helpful and beneficial. And if you haven't been using our Anki cards associated with these different videos, I'd encourage you to use them. If you have any questions for me, reach out to me. I'd be happy to help out. Share this playlist with your friends. Don't gatekeep it. And honestly, I don't know what I'm going to move on to next. I might start with the biology playlist or the lab techniques playlist. Either way, I'm going to be working on one subject after the other until we have a comprehensive MCAT playlist for each subject. Go and crush that MCAT.